Jai Hind dear students, I am Dr. Preeti Mittal and today we are going to continue with the chapter Cell, the fundamental unit of life. In the previous video, we have seen about nucleus, cytoplasm and the discoveries related to cell, and in this part of the video, we are going to discuss about the various cell organelles. What are cell organelles? Human body has many organs that combine together and helps us to perform different functions. Similarly, in a cell also, there are various small parts, small components of a cell, and they together help in the functioning of a cell. A cell is known as a basic structural and functional unit of an organism. Why is it called so? A cell is known as a structural unit because like bricks helps us to make a house. Similarly, cells together combine and form the whole organism. So a basic structural unit of an organism is a cell. Why is it called a functional unit? Every cell is capable of performing cellular respiration, that is breakdown of glucose to produce energy. Apart from that, every cell can perform excretion, that is removal of waste from it. Every cell performs digestion and nutrition, that is takes up the glucose, amino acids and use it for preparing the different cell components. And that is why every cell can be said as a functional unit because every function that is performed by an organism as a whole that could be performed by each and every cell as well. That is why we call a cell as a structural and functional unit. So now let's look about the functions and the structure of various cell organelles. Okay. Starting with the first organelle, that is endoplasmic reticulum. Endoplasmic reticulum, here you can see that it's present around the nucleus and it's a form of tubules. Long tubules are there. Long tubules as well as vesicles forms the endoplasmic reticulum. This endoplasmic reticulum is very important in a cell because it helps in transportation. Endoplasmic reticulum will be helping in transportation of material from nucleus to cytoplasm, from one part of the cytoplasm to another part of the cytoplasm when the things have to be transported. Apart from that, when the things have to be transferred from the nucleus up to the plasma membrane, then also this endoplasmic reticulum is going to help in the transport of material. There are two types of endoplasmic reticulum: rough endoplasmic reticulum and smooth endoplasmic reticulum. Rough word itself suggests that its surface is not going to be very smooth. But why is it rough? Rough endoplasmic reticulum is rough because of presence of small granular structures called ribosomes. Ribosomes are present over the surface of endoplasmic reticulum, which makes its surface rough. It's just like putting some sand over the road. Will that make the road rough? Similarly, these small granular structures are there that are called ribosomes, about which we are going to discuss more, and that makes the surface of this endoplasmic reticulum to be rough. And that is why a reticulum, endoplasmic reticulum that is containing these ribosomes is known as rough endoplasmic reticulum. If the endoplasmic reticulum doesn't has any ribosomes over its surface, then it's called as smooth endoplasmic reticulum, represented by SER, smooth endoplasmic reticulum. Okay, so endoplasmic reticulum we have seen it's of two types: rough endoplasmic reticulum and smooth endoplasmic reticulum. Now, this rough endoplasmic reticulum, I told you, it contains ribosomes, and ribosomes helps in the synthesis of proteins. Ribosomes are also known as the protein factory of a cell. So these ribosomes helps in the synthesis of proteins. And do you all know enzymes, hormones, as well as all the cell membranes, they all contain proteins. Apart from that, this rough endoplasmic reticulum is having the ribosomes, that is manufacturing proteins, and hence the one of the function of rough endoplasmic reticulum will be synthesis of proteins. What's the function of smooth endoplasmic reticulum then? Smooth endoplasmic reticulum helps in the synthesis of lipids. Lipids are also known as fats. So, lipids will be synthesized by smooth endoplasmic reticulum and proteins are synthesized by rough endoplasmic reticulum. Do you all remember while teaching plasma membrane, I told you all that plasma membrane is made up of proteins and lipids. And I asked you all to remember that. From where these proteins and lipids are coming, I hope now you are able to guess that. That is from the endoplasmic reticulum. So, proteins required for plasma membrane synthesis comes from rough endoplasmic reticulum and the lipids required comes from smooth endoplasmic reticulum. And that is why this endoplasmic reticulum, one of the functions is membrane biogenesis. It's very important as an individual question also, that what is membrane biogenesis. Formation of plasma membrane requires proteins that are synthesized by rough endoplasmic reticulum and lipids that are synthesized by smooth endoplasmic reticulum. And that is why endoplasmic reticulum helps in the formation of plasma membrane, which is known as membrane biogenesis. In liver cells, the smooth endoplasmic reticulum also helps in the detoxification of drugs, medicines, poisonous substances. So whatever medicines we take, that also will be detoxified by the liver by the help of smooth endoplasmic reticulum present in the liver cells. I hope that is clear. Apart from that, I told you that enzymes, hormones, they are also made up of proteins and lipids only. So these proteins and lipids are also used for the synthesis of enzymes and hormones in the body. We will read more about enzymes and hormones later on. Next is endoplasmic reticulum also provides a structural framework of a cell. What is the meaning of structural framework? Whenever there is a construction of a building going on, you must have observed that first some iron rods are fixed and around which the construction of bricks is done. So that framework is provided by those iron rods. Similarly, in a cell also, the basic function of maintaining a proper shape and providing a proper support to a cell is done by endoplasmic reticulum. It's spread all in the cell. It is also going to connect it is going to be connected even to the plasma membrane. So it's present all over the cell and so it forms a basic structural framework of a cell. Apart from that, if you microscopically observe the endoplasmic reticulum, its structure is similar to that of plasma membrane. So, the structure of plasma membrane and endoplasmic reticulum are similar. One more thing that you can observe is endoplasmic reticulum is having a single membrane. So, it's a single membrane bound structure. So, I hope all this is clear to you all. You are able to understand about endoplasmic reticulum. Now, I'll shift aside that you can take a screenshot if you want. And that's all about the endoplasmic reticulum. The next organelle that we are going to study about is mitochondria. Mitochondria is known as the powerhouse of the cell. So let's see to the structure of this powerhouse. Mitochondria is a double membrane bound organelle. It is having two membranes. It's a very special and a very important cell organelle. The outer membrane is porous and the inner membrane you can see that it has many folds. These folds increase the surface area. Why we need to increase the surface area? Because ATP synthesis is going to take place here. Why ATP synthesis? In our cells, every cell is going to perform cellular respiration. Cellular respiration means glucose will be broken down into its components, that is carbon dioxide and water, and energy will be released. This energy will be stored in the form of ATP, adenosine triphosphate. So, this process of breakdown of glucose to form ATP, and this ATP storage, it's done in the mitochondria. ATP is also known as the energy currency of a cell. Remember, it's very important. What is known as the energy currency of a cell? So that is ATP molecules, adenosine triphosphate. ATP is known as the energy currency of a cell. So this ATP synthesis is going to take place in the mitochondria, and that is why it is known as the powerhouse of the cell, because ATP is the energy currency, and whenever you are storing some energy, the place where you are storing the energy, that is called powerhouse. So mitochondria is known as the powerhouse of a cell. It's a very special organelle in the sense that it has its own DNA and own ribosomes. So it can divide on its own, it can synthesize proteins on its own. So very few organelles are there that that have their own DNA and ribosomes, and that is mitochondria. The other one we'll read in plant cell, that is chloroplast, that is also having its own DNA and ribosomes. All that much is given in your NCRT book, but I would like to tell you something extra from this topic. Actually, cell topic, especially the cell organelles, are very important for you all. In NEET also, you will be having some questions based on this topic. So it's really very important. And one more thing is there, you will be reading about cell once again in 11th standard as well. So it will be really better for you all that if you're preparing for NSO exams, or if you are preparing for some competitive exams later on, this will be useful for you to gain some extra knowledge. So here what I have written is that's given in your NCRT book. And now I'll be telling you something extra, not given in NCRT book, still it's very important. Okay. So, who discovered the mitochondria? Mitochondria was first of all discovered by Kolliker. Kolliker is a scientist who first saw the mitochondria, observed the mitochondria for the first time. But the name mitochondria was given by another scientist known as Benda. So Benda was the first scientist to give the name mitochondria. Mitochondria is a plural and mitochondrion is a singular term used for a single mitochondria. I told you that the outer layer is porous and it's continuous. The inner layer is folded, and these folds are known as cristae. And whatever matter is there inside this inner membrane, the inner portion, whatever is here, that is called as matrix. On the cristae, there are, you can see, some bead-like structures are there, and they are known as oxysomes. These oxysomes contains the enzymes that are required for ATP synthesis. So we have seen in NCRT also it's given that ATP synthesis is going to occur here. But how that is possible? Because of oxysomes which are present on the inner lining of the cristae, and that is having the enzymes for synthesis of ATP. That is why mitochondria is a site for cellular respiration. Okay, cellular respiration means every cell is performing respiration, that is breakdown of glucose to release carbon dioxide and water. Okay, this breakdown of glucose requires oxygen, so breakdown of glucose in presence of oxygen to release carbon dioxide and water, that is known as cellular respiration. Okay, so that's all for mitochondria. I'll shift to side so that you can take a screenshot. Our next cell organelle is Golgi apparatus. Golgi apparatus was discovered by the scientist Camillo Golgi. He was a great scientist. He even got a Nobel Prize for his work that he did for the staining of nerve cells. And the reaction that he carried out is known as black reaction, that helps to study nervous system by staining a neuron. So he got even a Nobel Prize for that. So it was a great scientist, Camillo Golgi, who discovered the Golgi apparatus. Now, what's Golgi apparatus and what's its function? So the structure first, Golgi apparatus is made up of a single membrane bound vesicles which are arranged almost parallel to each other in a stack. Now, what is a stack? Stack means one above the other. So, if you put a stack of coins, means putting the coins one over the other. That is called a stack of coins. So, the Golgi apparatus is made up of single membrane bound vesicles which are arranged almost parallel to each other, and it's like a stack, and that is known as cisterns. This stack of Golgi apparatus, this stack of vesicles is known as cisterns. So, that is the structure of Golgi apparatus. This Golgi apparatus is in close connection with endoplasmic reticulum. It's present close to the endoplasmic reticulum. That is also for a reason. Endoplasmic reticulum, you all know that it synthesizes proteins and fats. So, rough endoplasmic reticulum synthesizes proteins, smooth endoplasmic reticulum synthesizes fats. So, endoplasmic reticulum basically synthesizes proteins and fats. And the function of Golgi apparatus is the storage, modification, and packaging of all these products in vesicles and sending them to the places where they are required. We can understand the functioning of Golgi apparatus comparing it with a chips factory. So suppose our endoplasmic reticulum is just like a farm. Any factory, if you have to set up, that you prefer to set up to a place which is close to the area where the rare raw materials required for that. So if we talk about a chips factory, then the raw material is potato. So suppose the potatoes which are grown in this endoplasmic reticulum, and the chips factory, the Golgi apparatus, is going to store the potatoes. It's going to modify it into the form of chips and package that and send it to you. So similarly, the Golgi apparatus is close to the endoplasmic reticulum, that is close to the raw materials which are needed to be stored, modified, and packaged and sent to the areas of action. Golgi apparatus also performs the functioning of converting simple sugars to complex sugars. So that modification of simple sugars into complex sugars, that will also be performed by Golgi apparatus. Apart from that, one very important function of Golgi apparatus is the formation of lysosomes. Lysosomes, we are going to talk about that, but lysosomes are formed by the Golgi apparatus, because lysosomes also has some enzymes, and enzymes are made up of proteins only. So this Golgi apparatus will be helping in the formation of lysosomes and plasma membrane, which again is made up of proteins and lipids. So formation of lysosomes and plasma membranes will also be helped by the Golgi apparatus. I hope this is an easy organelle for you all to remember. I'll just shift aside so that you can take a screenshot. The cell organelle that we are going to discuss now is lysosomes. Lysosomes are a single membrane bound vesicle structure, and it is containing digestive enzymes that are prepared by rough endoplasmic reticulum. I told you earlier also that rough endoplasmic reticulum prepares proteins, and enzymes are a form of proteins only. So the digestive enzyme that is present inside the lysosomes, that is prepared by rough endoplasmic reticulum. Apart from that, remember that lysosomes are formed by the Golgi apparatus. Now, the lysosomes has a very important function in a cell, that forms the waste disposal system of a cell. Why it's said so? The lysosomes contain the digestive enzyme. So whenever any bacteria or some cell organelle is worn out, for example, if a mitochondria is worn out, it's damaged, so it has to be removed from the cell. So all such damaged cell organelles, any foreign body like bacteria entering inside the cell, that all will be removed by the lysosome. So it's known as the waste disposal system of a cell. It's also known as the suicidal bags of the cell. Suicide means killing oneself. And lysosomes are known as suicidal bags because if a cell is old, damaged, or worn out due to some reason, then these lysosomes are going to burst. And when it bursts, this digestive enzyme is going to spread into the entire cell and digest all the cell components. So the cell in which it was present, it is digesting that particular cell only, and so it's known as the suicidal bags of a cell. And it also helps to keep the cell clean by removing any waste, any of the dead material from the cell, and helps the cell to be kept clean. That is another function of lysosomes. That's all what we discussed right now. It's given in your NCERT book, but I would like to add something extra, not given in NCERT, but again important for your 11th standard biology, important for your NEET exams. So that is discovery. The lysosomes were discovered by Christian D. Due. D. Due was a scientist who discovered the lysosomes. And I told you it is having a digestive enzyme. What's the name of the digestive enzyme? That is hydrolytic enzyme. The digestive enzyme present in lysosomes is hydrolytic enzyme. And these are also known as scavengers in a cell. Why called scavengers? Scavengers means organisms which which eat up the dead and decaying matter. So they are also removing the dead cells, dead organelles, and so it's known as the scavengers in a cell. That's all about lysosomes, an important topic for your exams. So I'll just shift aside. You can take a screenshot. The next cell organelle that we are going to discuss now is present only in a plant cell and absent in animals, that is plastids. Plastids could be having some color, then they are known as chromoplast. Chroma word means color. So chromoplasts are the plastids that have any color in them, and if they don't have any color, that is they are colorless or white in color, then they are known as leucoplast. Wherever the word leuco comes, that refers to white color. So leucoplasts are the colorless plastids and the colored plastids are known as chromoplast. This chromoplast could be having different colors like yellow, orange. You must have seen the petals of the flowers having beautiful different colors, or you see different vegetables having different colors like brinjal. Whenever you see some fruits having different colors like red color of apple or orange color of orange, then that is because of the presence of chromoplast. But if the chromoplast is having green color, then it is giving a special name, and that name is chloroplast. Chloroplasts are the plastids that have the green pigment in them, and that is the chlorophyll containing plastids. This chlorophyll containing plastids are definitely going to help in the process of photosynthesis performed by the plants. That is why the chloroplasts are also known as the kitchen of the cell, because in the kitchen the food is prepared. Similarly, in the leaves the food is prepared in the chloroplast cells, chloroplast organelles. So they are known as the kitchen of a cell. So name the cell organelle that is known as the kitchen of a cell, that is chloroplast. Now if it's white in color, that is leucoplast, that is the colorless plastids. They are usually present in the roots and in the stem. In the roots sometimes, or in the stem, there is storage part of the plants, like potatoes. Potatoes are a modified stem only. Apart from that, sweet potato, that is a modified root. So they are the storage part of the plants where the food is being stored by the plants, and they contain the leucoplast. So leucoplast has performed the function of storing of starch. It could be oils, it could be proteins. So proteins, oil, starch could all be stored in the leucoplast. Plastids are a special organelles that have their own DNA and own ribosomes. Do you remember one more cell organelle that I told you it's having its own DNA and ribosomes? Think about it, recall that. Yes, that is mitochondria. So mitochondria and plastids are the only cell organelles that they have their own DNA and ribosomes. Apart from that, plastids that you need to remember is that they are a double membrane bound structure. They are having two membranes around them. So it's a double membrane bound structure. Now talking about something extra that's not given in NCERT book. First is about the term plastids was given by Ernest Haeckel. E. Haeckel you can remember, or Ernest Haeckel gave the term plastids. Apart from that, the structure of plastids is really important and interesting also. I told you it's a double membrane bound structure, so you can see the outer membrane and the inner membrane, both are continuous. Apart from that, inside the plastids, there is like a structure like a stack of coins. Stack of coins means coins placed one above the other. So that single coin is known as thylakoid. That single coin like structure is known as thylakoid. And these coins placed one below the other, that complete stack of coin is known as granum. Why is it important? Because in photosynthesis, you will read in higher classes, there are two reactions involved. One is light reaction and the other is dark reaction. So in NSO exams or in competitive exams, you are asked that where the light reaction of photosynthesis occurs. So the light reaction of photosynthesis occurs in the granum part of chloroplast. Now, where does this dark reaction occurs? So, dark reaction occurs in the matrix. Matrix is the filling. Whatever is there inside that space is there inside the chloroplast, that is the matrix. So, this matrix is having stroma. That matrix part is known as stroma. And that stroma is a site where there is dark reaction of photosynthesis. So, light reaction occurs in the granum part and dark reaction occurs in the stroma. And this is the organelle I told you earlier also, that it is having its own DNA and its own ribosomes also. Okay, so that stack of coins is there. A single coin is known as thylakoid. That complete stack of coins is known as granum, where the light reaction of photosynthesis is going to take place. The other structure is the matrix, that is the stroma, where the dark reaction of photosynthesis is going to take place. Okay, so that's all about the plastids. What you really need to remember is it's a double membrane bound structure having its own DNA and own ribosomes. It could be colorless, that is leucoplast, or colored, that is chromoplast. If it's colored and the color is green in color, that specially is known as chloroplast. Okay, the cell organelle which is known as the kitchen of the cell, that is also chloroplast. So I'll just shift aside, you can take a screenshot. Our next cell organelle is vacuole. Vacuole is present in a plant cell as well as in an animal cell. But in animal cell, it's very small, and in plant cell, it's really big. The plant cell have a big vacuole which stores amino acids, proteins, sugars, organic acids like citric acid in lemon and oranges. So these are known as organic acids that are all stored in a vacuole. The fluid present in the vacuole is known as cell sap. In case of plant cell, the nucleus is pushed to one corner of the cell because of the presence of a big vacuole. Whereas in case of animal cell, the nucleus is at the center of the cell because the vacuole is very small. The vacuole is also used by the plants to remove the waste from the plant cells. For example, the plant has to remove some waste, so it can store it in the vacuole of the leaves, and now when the leaves will be shed off, along with the leaves, the vacuole will be taking away the excretory products of the plants as well. So plants use the vacuole as a method of excreting out their waste products as well. In case of amoeba, the food is also stored in a vacuole. So, amoeba takes in the food through its cell membrane and forms a food vacuole around the food material. And then the food will be digested by amoeba. One more thing is there, that is rigidity and turgidity, is provided by this vacuole. If you see a fresh tomato, you see that the skin is very tense, it's tight, and that is only known as rigidity and turgidity. That is provided by the fluid content in the vacuole. In higher classes, you will read that cell sap vacuole, food vacuole are actually the types of vacuole. But in ninth standard, these types of vacuoles are not mentioned. One extra thing that I would like to add is the membrane around the vacuole is known as tonoplast. That is known as tonoplast. So that's all about vacuole. I'll just shift aside so that you can take a screenshot. Next to cell organelles are both membraneless, that is, they do not have any membrane around them. So the first one is ribosomes. Ribosomes are known as the protein factory of a cell. They help in the synthesis of proteins. Ribosomes are seen over the rough endoplasmic reticulum, found scattered in the cytoplasm, means here and there in the cytoplasm you can see many ribosomes. Apart from that, they are also present in mitochondria and plastids. So, the ribosomes are present on rough endoplasmic reticulum, mitochondria, plastids, and scattered in the cell cytoplasm. One thing that I would like to add that's not given in NCERT is the types of ribosomes. Ribosomes are of two types. 70S ribosomes are seen in prokaryotic organisms, and eukaryotic organisms have 80S type of ribosomes. Okay, so that's not mentioned in NCERT book. Next cell organelle is centrosome, about which in the NCERT book it's only shown in the diagram. Nothing is described about the centrosome in your NCERT book. So if you want to skip about it, you can skip that. But it's important for 11th standard biology as well as NSO exams. So I would like to tell you about it. Centrosome is made up of two perpendicular structures that are cylindrical in shape, and that is known as centriole. These centriole divide from each other, separate from each other at the time of cell division, and they go to opposite parts of a cell during the cell division. They play a very important role in cell division in case of animal cell. The centrosome is present only in an animal cell and absent in plant cell. So that's all about the different cell organelles. Now we are just going to discuss about the difference between plant cell and animal cell, and we are done with our all the cell organelles. I'll just shift aside, you can take a screenshot. Now let's discuss about the difference between the plant cell and animal cell, which is a very important question for your exams. The first difference is based on the size. A plant cell is usually larger in size as compared to an animal cell. The second point is cell wall. It is present in a plant cell whereas absent in animal cell. Third point is about plastids. We have seen about chromoplast, chloroplast, leucoplast. So plastids are present only in a plant cell and absent in animal cell. Vacuole. A large vacuole is seen in a plant cell, whereas a vacuole is small in size in case of animal cell. Next is nucleus. In case of plant cell, the nucleus is usually shifted to periphery, that is at one corner of the cell because of a large vacuole. Whereas in animal cell, the nucleus is usually at the center. So I'll just shift aside, you can take a screenshot. Now at last we'll see to some things to remember for competitive exams, NSO exams and all. So first is the organelles could be divided into three categories. Some organelles are membraneless, means they do not have any membrane around them, and that are ribosomes and centrosome. Second is single membrane bound organelles. Those organelles which have a single membrane around them, and that is Golgi apparatus, lysosomes, endoplasmic reticulum, and vacuole. You can remember it by that Golu loves eggs very much. Next is double membrane bound organelles, that are nucleus, that is having a double membrane, mitochondria, and plastids. one last thing that I would like to tell you is that human RBC, when it's in the mature stage, it doesn't has any nucleus, mitochondria or Golgi apparatus. So when it's getting mature, when it's developing at that stage of development, the human RBC has these organelles, but once it matures, when it's developed fully, then the nucleus, Golgi apparatus, mitochondria, they are all not present in a mature human RBC because that place will be occupied by a big hemoglobin molecule. Okay, so that is there in mature RBC in human beings, and not only human beings, in all mammals it's that. Okay, so you can take a screenshot if you want. Good day everyone.